Hi, I'm Philip Schlenoff, a high schooler from Tallahassee, Florida. For the last few years, I've been working on my project titled Antibody-Coded Magnetic Nanoparticles Targeting and Treating Cancer in a Dynamic Environment here at the Florida State University. Let me tell you a bit about it. The overall goal of my project was to create a nanoparticle-based cancer treatment capable of circulating through the bloodstream, specifically targeting cancer tumors inside the body, and then being heated externally to kill the cancer tumors while leaving healthy tissue intact. Now, researchers often run into a few problems when trying to approach cancer treatment this way, mainly getting these nanoparticles to circulate in the bloodstream. Now, that's why I'm so fortunate to work here at the Florida State University. Because a few years ago, researchers here developed a coating called SBS. Now, this special Zwitterion coating has a plus and a minus charge very close to each other. This neutralizes the surface of the nanoparticle, which allows these particles to circulate in the bloodstream without aggregating together, and it also masks them from the immune system, so you don't get any interference. So the overall structure of these nanoparticles can be broken down into 4 parts. The first is the magnetic iron oxide core. This allows me to heat the particles from outside the body using radiofrequency irradiation, which only targets magnetic material. This is coated by a silica shell. The silica shell helps biocompatibility and also allows me to add the final 2 ligands on, the first of which is antibodies. Now, these antibodies allow these particles to specifically target cancer tumors inside the body and not go to any healthy cells. The second of which is SBS, my special Zwitterion key. Once the particles have reached their intended target inside the body, radiofrequency irradiation can be used to heat the magnetic material inside the particles. And because the particles are only in cancer tumors, only the cancer tumors are killed, while the healthy tissue remains undamaged. These graphs here show that these particles are what I intended them to be. This first powder X-ray diffraction shows that the particles are a composite of iron oxide and silica, so they've got that magnetic material inside of them, and they've got the silica shell. The second graph shows that the particles are superparamagnetic, which allows me to target them with radiofrequency once they've reached the cancer tumors. This third graph here shows the stability of the coated particles versus the uncoated particles. The particles that aren't coated with SBS aggregate very quickly inside cell medium, which mimics the hazardous conditions of the blood. Oops. The uncoated particles aggregate very quickly. As you can see here, the absorbance has increased very highly, whereas the uncoated particles remain stable in the medium for a period of days. The second graph here shows that the particles are the intended size. They're small enough so that they can fit out of the pores in the fenestrated and the sinusoidal capillaries inside the body, which is how treatments like this work. They diffuse out of capillaries and they go into tumors. To test this treatment, I designed something called an animal simulation environment. I didn't want to just use a petri dish, because that's just kind of a static culture of cells. You know, it really doesn't mimic the way that a treatment would work inside a living thing, like a human or a rat. So instead, I designed something to mimic the diffusion of these particles out of capillaries and into tumors, the same way that a treatment would work inside a living thing. Cell medium is pumped through these tubes here by a peristaltic pump. Now, this is the same way that the muscles in the body pump blood. This first chamber here is basically just a control. I just want to make sure that the antibodies on these nanoparticles allow me to specifically target cancer cells on one side while leaving the healthy cells on the other side untouched. The important chamber is the second one. Before that, I've got something I call a capillary module. Basically, it's just a system of pores that the nanoparticles have to flow through in order to reach the second tumor, to make sure that the SBS coating makes these particles stable enough to reach their intended targets inside a living thing. So my results were very good. After just 90 minutes of radiofrequency treatment, almost half of these cells had died, 44%, and the control cells were unharmed. Now, this means that with optimization, with increased concentration, increased duration, maybe repeat treatments, this number can be increased significantly, and someday, hopefully, to 100%. I'd like to thank everyone who collaborated with me on this project. I worked under the supervision of Zeki Estefan. I'd like to thank Layal, Zahra, Raed, Joe Schlenoff, Amy Sang, and especially Sir Harry Croto for giving me the opportunity to give this presentation. So these are some of my references in case any of you want to look at any additional work.